Hello everyone. In this video, we will learn about the plant tissue culture. What is plant tissue culture? Basic terms used in plant tissue culture. Sterilization techniques. Culture media for tissue culture. Growth regulators. And about the micropropagation and its applications.
Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues or organs under sterile conditions on a nutrient culture medium of known composition. Plant tissue culture is widely used to produce clones of a plant in a method known as micropropagation. Explant is a small piece of living plant or animal tissue removed from an organism to initiate a tissue culture or for other biological purposes, such as grafting or research. Callus is a mass of undifferentiated mass of cells produced during the initial stages of tissue culture. Cell totipotency, the fundamental principle that a single plant cell possesses the genetic information and potential to grow into a complete, new plant, including shoots and roots. Organogenesis, it's the pathway where shoots develop from a callus, an unorganized mass of plant cells that forms from the explant tissue. Sterilization. In order to produce contamination-free clones, it's important to maintain microbial-free environment, and the technique of elimination of all kind of microbial contamination is known as sterilization. And the process of providing microbial-free environment is known as aseptic condition. It is important to maintain aseptic condition for successful tissue culture. Sterilization of glasswares. Glassware can be sterilized in hot air oven at 160 to 180 degrees Celsius for 2 to 4 hours. Sterilization of instruments. These can be sterilized by dipping in 75% ethanol followed by flaming and cooling. Sterilization of media. Media used for tissue culture is sterilized in autoclave at 15 pC at 121 degrees Celsius for 30 minutes. Sterilization of explants. Explants which were selected for tissue culture also need to sterilize before proceeding. First, selected explant must be thoroughly washed with water, followed by dipping it in detergent solution such as Tween 80 for about 5 minutes. To avoid microbial contamination such as bacteria and fungus, it is better to dip explant in antibacterial and antifungal solutions such as streptomycin and bavistine for 15 to 20 minutes. Then rinse the explant with water and surface sterilize the explant with 0.1% mercuric chloride solution under laminar air flow. Before inoculating it into the medium, cut down the edges of the explant, since it gets absorbed the chemicals, and wash with sterile water. Let's discuss about the culture media. As all culture needs nutrient for its culture, plant tissue culture also needs certain formulations for successful establishment of culture. Composition of widely used tissue culture media. 1. Macronutrients. As its name suggests, in plant tissue culture media these components provide the elements which are required in large amounts, concentrations greater than 0.5 millimole, by cultured plant cells. Macronutrients are usually considered to be carbon, nitrogen, phosphorus, magnesium, potassium, calcium and sulfur. 2. Micronutrients. It provides the elements that are required in trace amounts, concentrations less than 0.5 millimole, for plant growth and development. These include manganese, copper, cobalt, boron, iron, molybdenum, zinc and iodine. 3. Iron source. It is considered the most important constituent and required for the formation of several chlorophyll precursors and is a component of ferredoxins, proteins containing iron, which are important oxidation reduction reagents. 4. Organic supplements, vitamins. The only two vitamins that are considered to be essential are myoinositol and thiamine. Myoinositol is considered to be vitamin B and has many diverse roles in cellular metabolism and physiology. It is also involved in the biosynthesis of vitamin C. 5. Carbon source. This is supplied in the form of carbohydrate. There are various media formulated for various purposes in plant tissue culture with defined components that promotes the development of various plant organs. 1. White's medium. It was developed by P.R. White for the purpose of root culture. 2. MS medium. It was developed by Murshige and Skoog, which supports the induction of organogenesis. 3. B5 medium. This media was formulated by O. L. Gamborg for cell suspension and callus cultures. Nowadays, this media was used for proplast cultures. 4. N6 medium. This media was developed by Chu for the culture of cereal anthers. 5. Niche medium. This was developed by Niche and Niche for anther culture. Among all these media, MS media was widely using in plant tissue culture as it supports wide range of species culture. Since it contains all the essential nutrients required for organogenesis. Synthetic and natural media. When a medium is composed of chemically defined components, it is referred to as a synthetic medium. On the other hand, if a medium contains chemically undefined compounds, example, vegetable extract, fruit juice, plant extract, it is regarded as a natural medium. Synthetic media have almost replaced the natural media for tissue culture. Along with the macro and micronutrients, carbon and iron, tissue culture needs certain molecules called as growth regulators or growth promoters. In case of plants, there are mainly 5 growth regulators are there. They are auxins, gibberellins or gibberellic acids, cytokinins, ethylene and abscisic acid. Out of these 5, auxin, gibberellins and cytokinins are known as growth promoters since they promotes the plant growth, where other 2 are responsible for plant senescence or growth suppresses. Gibberellins. There are numerous, naturally occurring, structurally related compounds termed gibberellins. They are involved in regulating cell elongation and are agronomically important in determining plant height and fruit set. Only a few of the gibberellins are used in plant tissue culture media, GA3 being the most common. Cytokinins. Cytokinins promote cell division. Naturally occurring cytokinins are a large group of structurally related, they are purine derivatives, compounds. Of the naturally occurring cytokinins, to have some use in plant tissue culture media. These are zeatin and 2IP, 2-isopentyl adenine. Their use is not widespread as they are expensive, particularly zeatin, and relatively unstable. The synthetic analogues, kinetin, benzylaminopurine, are therefore used more frequently. Non-purine-based chemicals, such as subasti, cute phenylurias, are also used as cytokinins in plant cell culture media. These substituted phenylurias can also substitute for auxin in some culture systems. Auxins. Auxins promote both cell division and cell growth. The most important naturally occurring auxin is IAA, indole 3-acetic acid, but its use in plant cell culture media is limited because it is unstable to both heat and light. Occasionally, amino acid conjugates of IAA, such as indole acetyl L-alanine and indole acetyl L-glycine, which are more stable, are used to partially alleviate the problems associated with the use of IAA. It is more common, though, to use stable chemical analogues of IAA as a source of auxin in plant cell culture media. 2,4-dichlorophenoxyacetic acid. 2,4-D is the most commonly used auxin and is extremely effective in most circumstances.
Micropropagation. In vitro clonal propagation through tissue culture is referred to as micropropagation. Use of tissue culture technique for micropropagation was first started by Morrell in 1960 for propagation of orchids. Clonal propagation refers to the process of asexual reproduction by multiplication of genetically identical copies clones of individual plants. Technique of micropropagation. Micropropagation is a complicated process and mainly involves 3 stages, I, 2 and 3. Some authors add 2 more stages, stage 2004, for more comprehensive representation of micropropagation. All these stages are represented in Fig and briefly described hereunder.
Micropropagation of tissue culture begins from selection, nourishment of parental plant to the successful transfer of cultured shoot with or without root into the nursery. Micropropagation is mainly dependent on the totipotency of the cell. Stage 0. This is the initial step in micropropagation and involves the selection and growth of stock plants known as mother plant for about 3 months under controlled observable conditions. Stage 1. In this stage, the initiation and establishment of culture in a suitable medium is achieved. The most commonly used explants are organs, shoot tips and axillary buds. The chosen explant is surface sterilized and washed before use. Stage 2. In this stage, the major activity of micropropagation occurs in a defined culture medium. The propagation may occur through formation of callus or direct formation of organs. Stage 2 mainly involves the multiplication of shoots or rapid embryo formation from the explant. Stage 3. This stage involves the transfer of shoots to a medium for rapid development into shoots. Sometimes, the shoots are directly planted in soil to develop roots. In vitro rooting of shoots is preferred while simultaneously handling a large number of species. Stage 4. This stage involves the establishment of plantlets in soil, which is done by transferring the plantlets of stage 3 from the laboratory to the environment of greenhouse. For some plant species, stage 3 is skipped, and unrooted stage 2 shoots are planted in pots or in suitable compost mixture. Micropropagation mostly involves in vitro clonal propagation by 2 approaches. 1. Multiplication by axillary buds or apical shoots. 2. Multiplication by adventitious shoots. Organogenesis, the formation of individual organs such as shoots, roots, directly from an explant lacking preformed meristem, or from the callus and cell culture induced from the explant. Organogenesis is of 2 types, direct organogenesis and indirect organogenesis. Direct organogenesis. In direct organogenesis, the tissue undergoes morphogenesis without going through a callus or suspension cell culture stage. Indirect organogenesis. When the organogenesis occurs through callus or suspension cell culture formation, it is regarded as indirect organogenesis. Somatic embryogenesis, the regeneration of embryos from somatic cells, tissues or organs. There are 2 routes of somatic embryogenesis, direct and indirect. Direct somatic embryogenesis. When the somatic embryos develop directly on the excised plant, explant, without undergoing callus formation. Indirect somatic embryogenesis. In indirect embryogenesis, the cells from explant, excised plant tissues, are made to proliferate and form callus, from which cell suspension cultures can be raised. After all these complication steps, why this micropropagation is that much important? Let us discuss the Applications of micropropagation. High rate of plant propagation. The more number of clones can be produced from a single cell culture or from callus. Production of disease-free plants. Using plant tissue culture, the disease-free and disease-resistance varieties can be produced. Production of seeds in some crops. Some plants won't produce seeds naturally, so as cauliflower, onion. Through micropropagation, seeds can be produced. Cost-effective process. Compared to other techniques, micropropagation is cost-effective. Micropropagation is also helpful to clone and preserve extinct varieties.
Thank you for watching. Please subscribe. If you have any doubt, please comment down. If you need this lecture in the form of PDF, kindly comment down. And please let us know which new topic you wish to learn easily. We will cover it.